Okay, everyone, let's get started. So it's my pleasure today to introduce our very own Violeta Calleja-Solanas, who is a community ecologist working with both like theory and data approaches, particularly with interaction network and invasion graph types of data. And today she's finished her PhD in Mallorca and then come over to do postdoc here in Sevilla, and is going to talk to us about drivers of biodiversity loss under extreme drought. Welcome, Violeta. Thank you. I was too early.
Okay, thank you for the introduction. And as you might have read, I have added and more, because actually apart from this project that I have been doing here as a postdoc, I would also like to tell you a little bit more about what I'm doing. And in order to do that, I will need to introduce myself a little bit more in depth. So I've been here for almost 2 years as a postdoc. Maybe you have not seen me. That's because I'm in the green building. But before that, I did my PhD in physics. So you might be thinking, why is a physicist here? And okay, that's a legit question. But let's say that during my physics training, I realized that ecology was, this is from Cambridge Dictionary, I think. It's basically the science that deals with the general question of how living beings interact with each other and their environment. It doesn't say anything about the researchers that need to work in this problem or the methods that they are going to use. So I confidently thought that with my physics background, especially in the study of emergent phenomena and the tools that I have studied, like complex network, bifurcation, nonlinear dynamics, and blah, 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 I could also study one particular emergent phenomena, that is species coexistence. So I during my PhD, I humbly tried to answer the question of why are there so many species living together. But of course, as a physicist, my approach was a little bit of a spherical cow. For those of you that do not know the joke, basically, physicists are accused, and with good reasons, that when they approach their system of study, they would oversimplify the system. So if they were to study cows, the first thing that they will do is to assume that cows are spheres with uniform density and no friction. So, well, I started with study ecology, in particular competition, and basically in every paper that I have read about competitive interactions, they start the introduction with the complex paradox. So why there is no exclusion principle? Exclusion principle species? Well, you know what I'm talking about. Basically, because if species are competing, we can rank species in competition dominance. So at the end of the day, just the most competitive species, the strongest one will survive. But actually, this is not the case. And then you know that there's been several theories proposing mechanisms to overcome this exclusion principle. Yeah, species exclusion principle. Now I have remembered. And one of the approaches that really caught my attention was this of intransitive interaction. So basically, instead of assuming that there is also there is actually a competitor that is the dominant one, what if we just remove that assumption and we think that species are actually kind of playing a rock paper scissors game. So in a rock paper scissors game that all of you have played when you were little children, there is not a best strategy. So if species are playing this game, basically, for example, the rock species at some point would be winning, but in that period then the paper species would appear, and then it will decrease the abundance of rock species, but then scissor species appear, and so on and so forth. So basically, these dynamics allow coexistence through oscillations on species abundance. It seems that the problem is that oscillations are not, or cycles, that these interactions produce were not stable. So researchers started to propose additional mechanisms together with intransitive interactions to try to get a state with a fixed point. So, and in particular, this paper claimed that with introducing in our mathematical models of ecological communities higher order interactions, we were able to just get rid of the oscillations and obtain stable coexistence. The probably thought during my PhD that this approach had is basically that adding higher order interactions were really overcomplicate the system. So we thought there is not any other mechanism more ecologically inspired that can also get rid of these oscillations, and we thought about space in the sense that species are not entities in the void or in the memory of our computers, but actually they live in a space. And are only possible if the location of individuals is close enough. So during one chapter of my thesis, we studied these structure interactions and how they relate to coexistence in competitive community. We published it in actually a physicist journal. And basically our approach was to have a species embed in a space. So they have precise location that create one structure. Then these species can interact depending on one interaction radius that we were inspired in sessile individuals like plants that needs to spread their seeds. So basically this plant, the seeds of this little plant are going to compete with the seeds of the other plant individuals that are within the interaction range radius. If we vary this interaction radius, then we obtain an interaction network among the different individuals. And what we did was to start with different arrangement of species that the amount of structure that they had. So we started with random networks called RNG, is a typical model of networks, where individuals were placed at random and connected also to compete at random. Then here they are also connected at random, but interactions are just created within an interaction radius. And then we have a lattice that is a highly structured spatial arrangement that is basically like a grid or a cheese mat. And for each of the different structures, we varied the range of the interactions. So to narrow down, our question now was how does a stable coexistence is engaged with a space depending on the interaction range and the spatial structure in which we are embedding our individuals. The model, because we did just a simulation, is very simple. So basically here we have our networks, and at a certain point we randomly kill one individual, which leaves this an empty place, and that ignites competition among the neighborhood. We choose two random neighbors, and they spread their seeds, which compete for this empty place. And finally, the winner, in this case the blue species, reproduces. So the interaction is pairwise, no higher order interactions are involved, and also is intransitive because we choose to be so. And the results were that effectively the dynamics changed depending on the structure and the range of interactions. We were able to recover oscillations, cycles of species densities of several characteristics, and also one state that was actually a fixed point where species were coexistent but without oscillations, just small fluctuations around an equilibrium. In order to match the structure that produced each of these dynamics, we actually plot the trajectory in this structure that is called the simplex. Basically, its side is one species. So we realized that if species are cycling a lot with wide amplitude, then the area that is the trajectories are going to cover in this space is big, and if they are all just fluctuating around an equilibrium, then the area is going to be very small. So we plotted here the area of the simplets, small areas, equilibrium, large areas, oscillations, against the interaction range. And actually the mean degree, which is a proxy of the size of the neighborhood of our individuals. With the random graph with no structure at all, we actually didn't see any difference between long and short range interactions. But with the other two type of spatial arrangement that were spatially structured, there is a region here with short range interaction that actually is able to have an equilibrium. So now we have found the condition for this state to appear, and we want to know why it's appearing with that particular setting of interactions and graphs. And basically, since we are studying spatial interactions, the best thing to do is actually look at the space. So here you have the grid of the species for short range interactions and long range interactions. And we realized that with short range interactions, actually there were some clusters of species that inside those clusters, they were just individuals of the same species. So the individuals are dying all the time, but they are being replaced with individuals of their same species. And it's actually this mechanism of creating cluster, monospecific cluster, the one that reduces the interspecific competition, and finally it allows to get this stable equilibrium. And by the way, I said it's stable because we, of course, ran some tests and we perturb the system, see the scaling of the fluctuation, so we are confident enough to say that that state is stable. So basically the conclusion of this paper was that we were able to find a stable coexistence with an alternative mechanism to higher order interactions that was space. And in particular, when the individuals were spatially structured, located in the space, and they interact in a short range regime, we were able to obtain a stable coexistence. Then with a master student, we recovered the higher order model and worked towards a less spherical cow, and realized that if we added some ecological meaning in the model, in the sense that previously there was a neutral model, in the sense that the birth and death rates of species were completely the same. So if we start to add real vital rates, some variation in them, then the stabilization of the higher order just disappeared. Really, that it's super cool to study with maths and computers ecology, but if I really wanted to study interesting and meaningful questions in ecology, I needed to ask ecologists. So that's why then for my postdoc I, let's say, took off my physicist hat and put on my ecologist boots, or kind of, because with these floats, who knows? But basically I started almost 2 years ago to work with Oscar Godoy and Nacho Bartomeos. I have been doing modeling, but always with empirical data. So it was with data-driven questions, and in particular this one, we wanted to see what were the drivers of biodiversity loss under extreme drought. And to do this, they had such a terrific empirical data sets, because they've been collecting for, well, they and their entomologists and technicians and more PhD students and postdocs, a lot of people actually was involved, to sample through more or almost 10 years some sites that are in Doñana. We have here in red 3 plots of communities of wild bees, and then here there are actually 9 plots of annual plant community. these are, for example, some of our main characters. So we have different type of species of wild bees that were also annual, which is actually quite a good system to study these questions. And luckily enough, both wild bees and annual plants were subjected to the same environmental driver, what was precipitation. And not only that, but precipitation in the recent years has been way less, has been decreasing. So we could say that the anomalies in precipitation, according to the historical mean, are telling us that there is a drought period. So we wanted to know how does diversity changes during this period. And in order to answer this question, we again used this long-term data set across multiple sites with very well-resolved data, and we constructed a model. And the novelty of this model is that it's on context dependency, because now what we are doing is to make the species interaction and the intrinsic growth rate to be able to vary in time. Why? Because we have added precipitation dependence parameters. So we can feed our data to this model and obtain one snapshot of species interaction each year for each precipitation value, and this can allow us to stability and diversity through through the years. In order to study stability and diversity, we use the structural stability approach that basically is a set of concepts, a theoretical tool that allow us from the intrinsic growth rates and species interaction measure some more ecological concepts related to ecology, like for example, species coexistence opportunities. And here is when it came our first surprise, because if we plot these coexistence opportunities against the precipitation anomaly, so a negative anomaly means a drought period, we see that coexistence opportunities actually increased during that period. And regarding fitness differences, they increase or decrease during drought depending on our system of study. Green is for plants and orange for bees. So it was strange, and it was stranger that when we measure the overall structural stability of the system, we see that actually the interplay of these two previous results make the stability of the system actually not to change a lot through the precipitation anomaly range. So, I mean, we have a lot to explain here, don't we? So the first thing that we did was to realize that these points were actually averages between sites and species. So when we look at the particular species and sites, we discovered a lot of context dependency that drive these results. In particular, we find that the species are affected differently for the drought, and in what manner. So first, their persistence, easy to understand, but basically we have plotted a proxy for persistence. So if these dots are on the left side, so they are positive, that means that the species tend to have more persistence when during wet years, and the opposites. Negative means that more persistent when there is a very dry year. And we find that there is not a general tendency for the species of our communities. So a certain set of environmental conditions makes winners and losers. Some species will benefit and others definitely not. And not only that, but the sites in which each species is living also affects how it develops, in the sense that here we have one plant, and these lines are the intrinsic growth rates in different sites. And we actually see a huge variety, not only in the x-axis, but also for the sites. So the spatial contingency makes not all sites equally suited for the species. So basically, these anomalies in how species are affected by precipitation is what makes the fitness difference to vary a little. And okay, and then to explain this increasing coexistence opportunities, we analyze the interactions. So for each year, I have already told you that we obtain one interaction matrix, and if we looked at some properties of those matrices across the precipitation anomaly gradient, we see that the diagonal dominance, that is the importance of the self-limiting effects of the species, the intra-specific interactions and the inter-specific interactions, is modified across the precipitation range, and in particular here during drought, the diagonal dominance is smaller, which means that dry years actually lessen the stability effect that having large self-limiting effects provides to the whole community. That regarding the intra-specific interactions. Then with the inter-specific interactions, we found also a very interesting thing. Here we have plotted facilitation with this line, and here with a dotted line, the negative interactions. And I want you to focus on these two things. Here, during drought periods, there is an increase in facilitation, and here, and especially here, I mean around the mean, there is an increase. There is more increase in competition. So actually, increasing facilitation during hard conditions, that remind us to the stress gradient hypothesis. So our results were coherent with this hypothesis. And not only that, an increasing facilitation is also related to more coexistence opportunities. So this is our explanation for the increase in coexistence opportunity during the harsh years. The interactions year by year rearrange in a way that they can explain these coexistence opportunities. So with this variation and these results, finally what we wanted to see are the title of this part of the talk, the biodiversity loss. So we took each snapshot of our system through time. So we had one set of interactions and intrinsic growth rates, and simulated just to see what was the potential final state of the system if those conditions were permanent. And what we found was that this is the Shannon diversity index. So we found that up to a certain threshold of precipitation anomalies, we had quite a high Shannon diversity, but then there is some point that the system just disappeared. And when we looked at the composition, actually we realized that these compositions were very heterogeneous. We had one species, the orange one, for example, that is dominating, which is something not very good in the sense that the less common species may be more prompt to get extinct. And more or less the same pattern was found in the plant data set. So basically, even though the structural stability that we were able to measure didn't change with the precipitation anomaly, the potential communities that these anomalies were generating were predicted to be, here in the extremes, less biodiverse and also with an heterogeneous composition. So basically, the conclusions of this work for us, that is first of all that the story is complex. I know, I hope I have telling you in a proper manner. But there is a good point, that is that these results are common to both taxas, which might be important if we then want to, in the future when our research is really applied, to infer some some politics and laws about how we can conserve our communities during drought periods. No, sorry. No, okay. So basically the take-home message is that drought generates winners and losers between species. So this context dependency also generates interaction that rearrange according to the Strand-Harrian hypothesis, but decreasing self-regulation when then all of these causes that the potential future communities will be less diverse than the ones that we have right now. So then, apart from doing theoretical modeling, during this postdoc, I have been also lucky enough to combine the mathematical tools that I have in my background with empirical data, just to try to develop new theories to fill the gaps between ecological understanding. And in this case, I'm going to present you a work done in collaboration with mathematicians from the University of Sevilla. And actually we recover these cycles. And what we did was realize that there are different proposed building blocks for maintaining biodiversity. On the one hand, if we just focus on pairwise interactions, we can obtain an ecosystem assembly that is linear or hierarchical, in the sense that we start with an empty island and species are arriving one by one, and then the system, via invasion, the system finally is created. And on the other hand, we have other approaches, as the one that I have told you in the first part of the presentation, where coexistence was reached through intransitive cycles, for example, whether they were here with the three species coexistence as we saw in my simulations, or here in which just one species enters first the ecosystem. So if other species enters, whether it's going to die or it's going to dominate the previous species, and the community is going to cycle around these sub-communities. So with this work, we wanted to answer the question on what are the characteristics of these two regimes and the cyclic dynamics, and more importantly, to answer the question of why they are rare in nature, in the sense that I have not told you previously, but these intransitive interactions, even though mathematically, theoretically, and with simulations, they give very promising results, then if you go to look at them, it's not a consensus whether they are really there or not. In fact, the balance is more towards the fact that they are rare in nature. We have some examples of microbes, like Escherichia coli, and some lizards and corals that clearly have these intransitive, not very well spread. And in order to answer these questions, what we are going to try is to link this community assembly up to coexistence with the matrix of interactions that our community have. In particular, we found that these were connected to a matrix of interactions that had their intraspecific values higher than the interspecific, so the ones that are called diagonal dominant. And then these ones are related to other type of interaction matrices, are called Lotka-Volterra, Volterra-Lyapunov stable. Doesn't matter actually the definition, but basically, since we saw a connection between the type of interaction matrix and the final assembly structure, we created this ratio of asymmetries of the interaction. So basically, in order to classify this assembly structure, we decompose the interaction matrix in one part that saves the intra-specific values plus some baseline interaction that is symmetric for each pair of species, plus then the asymmetries that those species have, in the sense that mutualistic partners. So maybe the benefit that the flower obtains being pollinated is not the same as the effect that the pollinator obtains for pollinate. So these things. So we just defined this ratio, we call it asymmetry ratio, between the strength of the asymmetric part and the symmetric part. And first of all, when we look at those cycles in the assembly graphs, depending on the asymmetry ratio, we realize that there is a region here where they are quite probable that correspond with asymmetry ratios more towards the asymmetric part of the spectrum, and then here other networks were not just that asymmetric, where the probability was absolutely 0. And not only that, also the more possible species that we have in our species pool to invade, the more probable is to a cycle to appear. So it's supposed that species-rich communities with not 7 species but 100 would have, in theory, a lot of cycles. But why are those cycles commonly known in empirical systems? Well, we could also be able to discover more type of cycles that, again, were never studied, neither seen before. And the question remains why they are not seen before. And these cycles are multi-species cycles and species variant cycles. These are the cycles like in the rock, paper, scissors game or rock, paper, lizard, spoke game or whatever. So one subcommunity of one species and goes to another subcommunity of another species and so on. But in multi-species cycles, we can have communities with the same. By the way, this is an error because I have here some numbers repeated, but basically we have cycles among subcommunities that have more than one species, so actually real communities. And then we have species variant cycles in which the subspecies involved can have a different number of species. Here, for example, species 1 and 3 successfully invade the subcommunity of species 4. So they allowed for simultaneous invasions, which actually is something that we can think will be also probable in nature, that 2 species arrive almost at the same time to our hypothetical island. And what's more, for now, the results that I have shown you were done with simulations where the intrinsic growth rates of species were all equal and 1. I know this is another spherical cow, but we did it just to isolate the effect of interactions. If we turn on the variability that we know it exists in nature of the intrinsic growth rate, this is this axis, we obtain that there is just this region with a high probability of cycles. As we go up in each of the vertical lines, the probability of cycles decreases. And not only that, if we look at now the number of nodes basically are the number of subcommunities in each assembly graph. So intuitively, the more nodes you have in an assembly graph, the more possible paths you have towards coexistence. And here is the contrary. There are less and more poor assembly graphs with a lot of asymmetric ratio, but the variability of intrinsic growth rates again decreases these possible graphs. And actually, with these results that I have presented to you, we were confident enough to propose an explanation of why cycles are rare in nature. And basically, this is because if we focus in this graph, nature is here. So within nature, the asymmetry ratio of the interaction matrices that we found in nature is less than 1 in the sense that we have empirical that the self-limiting effect of a species usually are greater than the interspecific one. And then also the variability of intrinsic growth rates usually is high instead of low. And just in order to prove it, we found here 21. If you have more, you are welcome to bring it to us. 21 empirical matrices of plant communities. From those, actually just 9%, what is 2 species, two communities presented cycles. All the other ones, there were no cycles at all, and were exactly in this part of the plot. So they had an asymmetry ratio less than 1. So basically the conclusion of this piece of highly theoretical but also with some data work is that we proposed this asymmetry ratio as a simple metric for cycles in ecology. And we were able to find and classify more types of cycles than the ones that were thought to just be. And our explanation about why cycles are rare in nature is because in nature we do have differences in species growth rates, and because there is a tendency towards a domination of self-limiting effects. And thirdly, that's all that I wanted to tell you. So let me just do a brief announcement. If you are interested in the second work that I have presented, we have an introduction to a structural stability workshop in Pontevedra this summer on Monday, the whole morning. So you are kindly welcome to join us if you are interested in knowing more about this approach. And that was all, so thank you very much.
Thank you, Violeta. We have time for questions. Anyone want to kick off?
Yeah, thanks for your talk. Well, what the thing I could understand more or less in the first section, you conclude that one of your main discoveries that the facilitation that allows coexistence opportunities has been produced in 2 taxa in plants and pollinators in bees, and you say okay. But both groups are in some way related. They are not entirely free because they are interacting organisms. What's your opinion if you could select another clearly independent taxa in order to interact between them? For instance, I don't know, snails and bees, or plants or birds, not associated to that plants in some way.
Yeah. Well, first of all, even though of course these 2 taxas are related, the 2 different data sets that we work with were not related, in the sense that we didn't sample the plants in the same place at the bees. So in that sense they are uncoupled. But then I admit that there might be some correlations, but this is not a bad thing, in the sense that as far as I know, the stress-gradient hypothesis was firstly developed for just plants. So just going one level up and saying, no, if there are other taxas that may or not may be related with plants that also have it, is a good step forward. Of course, the next step would be to find this hypothesis also proved in taxa that has nothing to do with plants. And yeah, I think that in other taxa that have some mutualistic partnership, it will also appear. Yeah.
Thank you.
Violeta, how different are the networks over years in terms of species composition and also on the interaction pattern in the first part of your talk?
Okay, there are some other measurements that we have performed in the matrices that I have not shown. But for example, the temporality of the networks that basically measure from one snapshot to the other. How we see that year by year they were quite difference, but always the same difference. And what really mattered was the anomaly. So if we rank the matrices according to their similarity, I know similarity has another meaning in networks, but if you want, according to the years, they were always more or less, they had the same difference. But if we shorten them according to the precipitation anomaly on each year, then the more precipitation anomaly, the more different in composition and structure they had. I don't know if that answered your question.
You compute the structural stability for each for the network each year, right?
Yeah.
So would it be possible that the fact that you don't see any difference in structural stability among years be a consequence of the network over time more or less is the same?
No, in that sense, it's not the same. So some years we have some species turnover. We maybe okay, it's super tiny, but great differences between links. There are some that are present every year, but others are completely different, and also the weights are different in the sense that some years they are positive and other years are negative. So yeah, we quickly discard this idea that the structural stability was the same because of the networks were the same. No, they are not. I don't know if you can come up with a good measurement to prove it. We have measured the temporality, and we were happy with that. But if you have any other suggestion, it's very welcome. This noise. Okay. Vale.
Okay. Any more questions?
Super cool work. I found the pattern of seeing increased kind of number of species with drought until you hit a threshold really interesting. And I was wondering if there was a way of also quantifying kind of the strength of species, like the competitive effects. Like, is it that there's a reduction in competition, and then at some point they hit physiological thresholds? Can you put numbers to that, or did you? And okay, and then I just totally missed it.
The variation of the weights of facilitative and negative interactions actually is continuous. Yep. So here in the weights we are not able to see any abrupt transition from some precipitation anomaly to another.
Yeah, interesting. Can you do it for physiological tolerance too, though? Like, so basically on the just whether or not the plant or the bee can tolerate.
Boof. I don't know. I mean, I'm not confident enough to answer that question because we have not looked at that thing. But yeah, instead of saying something random, I will just say that I'm not that sorry.
Cool. Thanks. It was a great talk.
What means a cycle and how we should study cycles in nature in terms of...
Wow. Okay, books could be written about that question. Okay, so first of all, I mean, the work of the cycles started because of this paper of 1975 of May and Leonard that is with these papers also by May alone in the 72, yes, about how simple models in the sense that they have very simple equations with 2-3 species, can actually give very interesting and weird behavior, even chaos, yes, with 3 species. So in this paper, they discovered that under a certain parameter range, you obtain these cycles, and they think, well, this is very unrealistic. Let's move on. But then there has been this body of literature about intransitivity, and actually May and Leonard model, I think it was intransitive. Yes, no, I don't remember. But so basically, there are a lot of people working on these kind of intransitive systems, but then there are not that much empirical evidence that they are really there to us to see them. So basically, with this, we wanted to answer the question of why is that. And then also, while we were doing this work, we realized that we ourselves were confunding several types of cycles, in the sense that one thing are cycles that we can find dynamically in the system, in the sense that I go to the field and measure abundances of foxes and hares, wolves and lynx and rabbits, and we see that they have oscillating populations through time. So this is a dynamical cycle. But then we have these cycles that we could call assembly cycles, in the sense that we have community that then disappears and other community appears, and this disappearance and appearance of community is cycling through time. And this is another mechanism, and with our framework of disassembly graph, we are able to look at these kind of cycles. If not, we can talk more later.
Thank you. So thanks, Violeta. And about this slide, so you quantified interspecific interactions, right? And these are observational communities, or are empirical communities, or? So
what do you mean by observational communities?
It is not non-experimental communities.
No, they are real.
So you went to the field and you quantified the interactions there.
Yeah, well, I didn't go, but yes. No, we did not quantify the interactions. We measured abundances. So our initial data set is 1 year, one site, abundance of species.
Okay.
So then, since there are several sites, and I mean, for physicists it's not a lot, but 10 years actually, now I have realized that they are a lot in ecology. So it's long-term data set. Then we have inferred the interaction values.
Okay.
So it's the model that gives us the interaction values. We do not assume that a pair of species is interacting, and we do not assume neither if the interaction is positive or negative. We do not assume anything.
So you are, but my question is, how can you infer? How did you infer positive and negative interactions, facilitation and competition? Are those interactions based on coexistence, co-occurrence patterns between the species? So if two species have many individuals within the same space, they are facilitating each other, or? No,
it's an inverse modeling technique. So you have a time series of points, and we particularly use generalized linear mixed models. We feed it. We are aware about doing this, but the errors were not big, and the goodness of feed were quite good. So it was like if you want to feed points to a line. So it's this procedure, but more complicated. So the fact that one interaction is for a certain value and a certain thing, basically is because this is the value that better reproduces the abundance of that pair of species.
Okay, thank you.
So then, how did you measure fitness, and what the values of fitness that you plot in the figure mean? Is the average fitness community?
I didn't go into deep in the methods of how we measure that, because they are not very intuitive or suited for a talk, but basically with the structural stability approach, we measured the fitness. Are you familiar with that? Okay, so basically interactions define a region where species can feasibly coexist, but then whether they are in that region or not depends on the intrinsic growth rate of those species. So basically in that framework, every year actually, since our intrinsic growth rates are changing, we have a point that is moving in that space, and also the shape of and size of this region where species coexist also moves and varies. Okay, so then the fitness basically was the distance for each species to the region of the space in which it is predicted that species will go extinct. So we so it's at this distance. So basically it's telling if there were a random perturbation, how in intrinsic growth rates, how big that perturbation should be to extinct that species according to this framework.
So, and what you plot is the average value for each species?
In each site, yes.
So this is a plot for
this one?
Each point is a network in one place, one site, right?
Is the average over the networks and the sites, because
okay, so each point is a site. The average of the networks at that site
is a year for our dataset that has been averaged over species in each site, so also over sites. Sorry if I
see.
Yes, basically each point is a year in the whole sense.
Yeah, thank you. So I'm a bit confused about, for example, the stability concept. Basically because stability doesn't exist in nature, right?
Oh
everything
yeah, right,
is
completely
moving,
agree.
everything is changing. But your understanding or your interpretation of stability is like staying put.
What, sorry? Staying
put in a site. So like keeping interactions, keeping relationships, keeping something.
No, not really.
So because as I see stability, it's not the ability to stay the same over time or across the space, but it's the buffering ability of a species against environmental disturbance.
Yeah, precisely the coexistence opportunity, what we call coexistence opportunity, actually is precisely that. Is the amount of perturbation the system can tolerate in its parameter until some species go extinct. So yeah, with stability, I do not mean by any means this fixed point with stable densities that I studied in the first part. It's not that. Species can fluctuate in densities, of course. They can go very big, very small, but the thing is that they are in a parameter region that is safe enough in the sense that if the parameters change because, for example, precipitation anomalies, the species is going the richness.
Right. Well, that's good because the mechanism explaining that buffering ability is actually interspecific variation, genetic variation within a species, which is able, to play around with the environmental perturbances. Are you taking that into account? Because that would be the mechanism explaining these situations.
You mean adding evolutionary?
No, meaning that systems that are more variable, so showing different phenotypes and showing different possibilities to interact with other organisms, they will buffer the effect of environmental variation like droughts that are quite or are not variable. So they don't have the chance to react to environmental variation. So for ecologists and population biologists, that's the key of the whole thing. So the more variable you are, the higher the fitness you will show in a system. Have different cards to play in different games.
Yeah, definitely this is another side of the fitness, but this framework as it is now is just at the species level. So maybe, of course, this species variation is the mechanism behind the ability for these perturbations, but within it now, we cannot explain it that in these terms.
But if you change just for a while of a species as a frequency distribution of traits, instead of something that we give a name, but it's a frequency distribution of traits, you will see, that then, you have many options to play with these kind of things. So, and there are a few examples here, recent theses about this kind of topics that can be very interesting to talk about.
Okay.
But for me, variation is everything. And this is why, I'm a bit shocked. But anyway, thank you.
Okay.
Yeah, I'm going to be short. Thank you, Violeta. Very nice talk. I have two comments. One is for this slide. I don't think you have included any temporal autocorrelation in the models, but this might exist because even for your graph showing the drought, I mean, you were showing like a trend towards more drought over time. So the aridity is each year is bigger because the previous year was also very dry. So you have like a temporal relationship between years for the drought, and also for the biotic communities, you might have also a temporal correlation, because if some species were more abundant one year, they're going to have more seeds, and the following year they have more probability, more likelihood to be also abundant. So I think you should somehow include any temporal aspect in the models if it's possible.
Well, right now I'm very happy that despite the limits in the data, we have been able to correctly fit the models. But yeah, this is an improvement that could be made. But also remember that species are annual, so the data that they are going to have would be in the seed bank if they have one. But yes.
Yeah, the species are annual, but you have the seed bank. So the previous year seed bank is determining the next year. And then, just for a maybe possibility, as I understood, you are not including bipartite interactions, right? Just only the plants and the bees.
Yes, exactly.
Yeah, but I think you have the data there, no? Knowing you have proven that when the year is drier, you have more species coexistent, but you also have more interaction between these species, between the bees and the plants. So these kind of questions are super cool also, because we also think that the stability of the system is also in these bipartite interactions. So maybe including that for the future. I know that it's not the problem now, but for the future could be super cool, I think.
Yeah, thank you for the suggestion. I think in the wild bees dataset that in principle could be done, but I do not know exactly how the plants have been collected there. But then keep in mind that one thing that precisely wanted to do is just not to suppose that there were a bipartite or any kind of a structure there. We wanted to see the nature of the interactions, not the other way around. Yeah, thank you.
I think with that we thank Violeta again and see you next week. Thank you.